Welcome to the fourth video for Chapter 2. Section 2.4 focuses on different types of staining, which will be very helpful in the first few weeks of lab. Most microbes are colorless or transparent in their natural state, so we need to do something to improve their contrast and visibility before we look at them under the light microscope. And there are two main ways to prepare your sample. One, the easiest way is a wet mount. You take your slide, you add your specimen onto the slide, and then you either put a drop of liquid or maybe stain on top, and then you place a cover slip on top of that before you view it on the microscope. The second way is you might have a fixed specimen that you need to look at, and this means fixation means you're going to stick the cells to a slide, attach the cells to a slide. You can do this through heat fixation or chemical fixation. In both ways, you're going to attach and preserve your specimen, but they work differently and they work how they sound. In heat fixation, you usually smear your sample on a slide, you let it air dry, and then you put it over a heat source, which will kill the cells and also attach them to your slide. This allows for preservation as it stops movement and usually preserves the structure of the cells. Chemical fixation is similar, but instead of heat, we're going to use some kind of chemical. And you might have encountered these in your other classes if you've ever looked at tissue samples in jars, or maybe you worked with a cadaver. So common chemicals are things like formaldehyde, also known as formalin, methanol or glutaraldehyde. Chemical fixatives are really good at preserving the overall shape and delicate structures of cells and tissues. So they're often preferred over heat fixation for histology, for tissue preservation. Here we have two examples of heat fixation. The first one on the left is a slide warmer. Then we have a microincinerator in the middle. The last one on the right is chemical fixation. So it looks like they have a tissue sample in formaldehyde, also known as formalin. And this will kill the microbes, or in this case, the tissue sample, kill the cells. That stops the degradation, the breakdown of the tissues, and preserves their structure so you can look at them at a later time. So in addition to fixation, we almost always need to stain our sample too to increase contrast or to enhance certain features of the cell. So stains or dyes are salts. This means they have a positive and a negative component. And one of the components will be a colored ion that we call the chromophore. The other charged component will be called the counter ion. So if the chromophore is positively charged, it's going to be a basic dye. And common examples we have in lab are crystal violet, methylene blue, we'll see malachite green, and safranin. If the chromophore is negatively charged, it's going to be an acidic dye. Things like acid fuchsin, eosin, nigrosin, Congo red, we'll see these in lab. Positive stains are going to bind to cells, and they're going to make them colored against a clear background or a white background. Negative stains are usually repelled by our cells, and they're going to stain the background. You'll see the slide or the background is stained, but the cells themselves will be transparent. The reason for this is because cells overall are negatively charged, right? The phospholipid membrane is negatively charged overall, proteins in that membrane are negatively charged, and in bacteria, the cell wall is negatively charged. So most of the time, basic stains will bind to the cell. Okay? Acidic stains will repel the cell and bind to the background. And here I have a really, this is actually methylene blue, where this part is the chromophore, and you can see in this case it's positively charged, it's basic, that's why it's going to bind to cells and stain them. This is the counter ion to stabilize it as a salt. On the left we have cell stain with crystal violet, which is a basic stain. Remember in chemistry, bases accept protons, and that's why they become positively charged. This positive charged stain will bind to negatively charged cells. On the far right, we have some kind of negative acidic stain. Remember, acids release protons and become negatively charged. Cells are also negatively charged, so they're going to repel each other. So that stain binds to the glass slide, which is positively charged, but not to the cells. In the middle, we have a typo here in the book. That should say a negative acidic stain. And this is interesting and kind of shows us that in some cases, acidic dyes can actually bind to certain tissues. And a lot of the times it's due to the specifics of the tissue or maybe the pH of the environment. There are two types of staining techniques you can use in lab. One is simple staining and the other is differential staining. In simple staining, you're just using a single dye, maybe crystal violet, and that's going to color everything on your slide the same color. So even the structures within the cell will all appear the same color. Differential stains are a little bit more complex. You're going to have multiple steps involving different types of dyes, and that's going to help you either distinguish between different types of organisms or different structures within an organism. These are all examples of differential stains, and we're going to go through each of these. All right, checkpoint time. First one, why is it important to fix a specimen? We need to fix the specimen to attach it to the slide. It also kills the microorganisms and preserves the structures within the cell. It also prevents movement of the organism. It prevents decomposition, so you can have a more accurate observation. Second one, what should be chemically fixed instead of heat fixed? So remember, for tissue samples or anything that's really delicate or heat sensitive, we should use chemical fixation. That preserves the structural integrity and avoids heat-induced damage. Third one, why might an acidic dye react differently compared to basic dye? Remember, acidic dyes, those have negatively charged chromophores, whereas basic dyes have positively charged chromophores. And remember that most cell surfaces are negatively charged, so basic dyes will be attracted and bind to the cells. Acidic stains or dyes instead, in contrast, are usually repelled, right? They're negatively charged, they're going to repel the surface of cells, so instead they stain the background, and that's also called negative staining. There are exceptions though, like that central picture I showed you earlier. If a specimen for some reason has positively charged components, it is possible for a negative acidic dye to bind to it. All right, the fourth one, difference between positive and negative stain. I kind of mentioned that earlier. Positive stain, the dye will bind to and color the specimen, whereas the background is not stained. Whereas in a negative stain, the dye is repelled by the cell or the specimen and stains the background. The last one, difference between simple and differential staining. Simple stains are usually just one step. You're just using one dye to color everything the same color. In differential staining, you're using multiple dyes to distinguish between organisms or between structures within the organism. The gram stain is a differential staining technique that's probably considered the most important and most commonly used technique in the micro lab. So this differential staining method allows us to classify bacteria into two main groups, gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria will appear purple after the gram stain, and this is going to be because they have a thick cell wall, whereas gram-negative bacteria will appear pink like these or maybe over here after the gram stain because they have a thin cell wall. So it differentiates bacteria based on their cell wall composition, and this was developed by Hans Christian Gram in the 1800s. There are 4 basic steps to the gram stain, and I'm going to walk you through the 4 steps in the next slide, but essentially they are crystal violet, the primary stain, Gram's iodine, the mordant, decolorizer, which is usually alcohol, and then safranin, which is the counter stain. The gram stain begins with our primary stain, crystal violet, and crystal violet is a basic stain that binds to all cells regardless if they're gram-positive or negative. And that's because cells, remember overall their surface is negatively charged, the stain will bind to the cells. In step 2, iodine is a mordant, and a mordant is used to stabilize stains or dyes. In this case, iodine traps crystal violet inside the cells. And this is because it binds to crystal violet, producing these large crystal violet iodine complexes, kind of like crystals, that clump and stay within the cells. So nothing much changes yet. The third step is the decolorizer, which is alcohol, and this can be ethanol or maybe an acetone ethanol complex. And what will happen here is in gram-positive cells, they have a really thick cell wall made of a chemical called peptidoglycan. Their thick cell wall is going to resist the alcohol step, so nothing happens. But in gram-negative cells, they have only a very thin peptidoglycan layer, and that is easily broken, easily washed away with alcohol. So when the cell wall is washed away, that crystal violet iodine complex leaves the cells. And these cells end up colorless, as you can see, after the alcohol step. In the last step, we have our counterstain, safranin. Safranin is also a basic stain, and it stains both types of cells. But because the purple stain was retained after the previous step, purple is darker than pink, so you don't see the pink. Purple masks the pink color. In contrast, for gram-negative cells, which were previously colorless, you will see the safranin, the pink color. At the end of the gram stain, gram-positive bacteria are purple, gram-negative bacteria are pink. And here they arbitrarily chose spheres here and rods here, but it's not true that they're always going to be purple spheres and pink rods. It could be vice versa as well. So you do have rods that are purple and spheres that could be pink. And this depends on the species of the bacteria. A few common errors can arise when you're doing the gram stain, and both of these usually affect gram-positive bacteria, making them appear pink or gram-negative due to two main reasons. One is if you use old bacteria, old cultures. So even though gram-positive bacteria are supposed to be purple because of their thick peptidoglycan cell wall, when they get old, their cell walls become more brittle, become more fragile. And when you add alcohol to old cell walls, even though they're thick, they can break more easily, washing out that crystal violet and allowing these cells to appear pink, even though they're actually gram-positive. Another common error is too much alcohol, too much of that decolorization step. So even if you have a thick cell wall, if you just apply too much alcohol or alcohol for too long of a period of time, that can ultimately still break down their thick cell wall, washing out the crystal violet and allowing or resulting in a pink appearance, a gram-negative appearance. So the gram stain is super useful clinically. It helps us classify pathogens. If you have an unknown sample and you're trying to figure out what it is or what it could be, doing the gram stain really helps you narrow that down. What's also interesting is that gram-negative bacteria are often more antibiotic resistant than gram-positive, and it's kind of counterintuitive. A lot of students will think that the thick cell walls will make them resistant, but in fact, the gram-negative bacteria, interestingly enough, even though they have a thinner cell wall, these bacteria also have an outer membrane. So they're going to have a normal plasma membrane, and then they have their thin peptidoglycan wall, but outside of that, they have another lipid membrane that helps them resist antibiotics. Let's do a quick review. So what's the role of Graham's iodine? Remember, iodine is the mordant. Its purpose is to bind with that primary stain, crystal violet, to form a crystal violet iodine complex that is larger than crystal violet alone and traps the complex inside that thick peptidoglycan wall of gram-positive bacteria. For the second one, explain the role of alcohol in the gram stain procedure. Remember that alcohol is a decolorizer, and in gram-positive cells, there's not much effect. It might dehydrate that outer wall, but in gram-negative cells, it's going to dissolve the outer membrane, break down the wall, allow the stain to wash out. What are the final colors? Gram-positive cells will be purple. Gram-negative cells will be pink after the staining procedure, and this means after all 4 steps. Going back to our clinical focus problem, the lab tech decides to do a gram stain and gets these results. So I see purple cells, that tells me they must be gram-positive, and their cell walls must have a thick peptidoglycan layer. Our second type of differential stain is the acid-fast stain, and this technique is used to identify acid-fast bacteria, such as those from the genus Mycobacterium or the genus Nocardia. Genera is the plural of genus. Acid-fast bacteria are a subset of gram-positive bacteria. Not only do they have that thick peptidoglycan wall, but outside of that wall, they have an additional waxy layer made of mycolic acids that resist normal stains. So in this case, we have to use a special primary stain known as carbofuchsin. The decolorizer in this case is acid alcohol, which is going to remove the stain from non-acid fast cells. So these are going to appear clear, but the acid fast cells will stay this color, which is kind of like a reddish pink. The third step, the counterstain or methylene blue, is going to color the non-acid fast cells since they were clear previously, but not the acid fast cells, because remember, they do not retain regular stains. They're going to have carbofuchsin still. There are two methods that are commonly used for acid fast staining. The Zeil-Nielsen method uses steam or heat to drive in that carbofuchsin, whereas the Kenyon method is going to be the cold staining technique. We don't use heat, and this is the one we usually practice in our lab. The reason we use the second method, the cold technique, is because using heat is great at producing a strong stain, but there are aerosols and it can be more hazardous. It puts more chemicals into the air. It could potentially put more aerosols of bacteria as well. In the end, acid-fast bacteria will appear pink or red, right? These are positive cells. Negative or non-acid fast cells will appear blue because of that methylene blue. And this is important in diagnosing certain diseases like tuberculosis, which is caused by the bacterium Mycobacterium tuberculosis. This picture comes from a different text, but I like how it contrasts gram-positive and negative bacteria, where you can see that thick peptidoglycan layer compared to the thin, and how gram-negative have that outer membrane that helps them resist antibiotics. Acid-fast bacteria, remember, are also gram-positive, so I don't really like this part that much because it should be a thick layer, but it shows nicely the outer layer of mycolic acid. And here are the steps again. Remember, we have the primary stain, carbofuchsin. It's going to stain both cells. The acid alcohol is going to wash away the carbofuchsin in non-mycobacteria or non-acid fast bacteria like Nocardia as well. And then methylene blue will stain non-acid fast cells. So in the end, acid-fast bacteria are pink, non-acid-fast are going to be blue. Acid-fast stains are useful because they help us differentiate bacteria with that waxy mycolic acid-rich cell wall. These identify acid-fast bacteria like Mycobacterium tuberculosis, and these bacteria don't stain well with normal stains, right, with typical techniques. So this is an important diagnostic tool for clinically significant diseases like tuberculosis, and later we're going to learn leprosy too. Remember, at the end, you're going to get red or pink cells if they're acid-fast, and blue cells if they're non-acid-fast. So acid-fast staining can be used to detect and diagnose tuberculosis in patient samples. You usually take a sputum sample from the patient and you apply that Zeil-Nielsen stain, those staining methods. If you get red or pink, then you culture that sputum sample further to confirm and identify, make sure that acid-fast bacteria are actually present. And Dr. Moore, who's shown at the bottom right here, she was a professor of bacteriology at Howard University in Washington, D.C., who actually pioneered many of these methods for tuberculosis research. Another way to diagnose tuberculosis to make sure these bacteria are present is through the use of antibodies. Remember that you can label antibodies with fluorescence, and this is known as immunofluorescence microscopy. These antibodies will be specific to tuberculosis bacteria. So if you see fluorescence, you can be sure that these bacteria are present. So far we've talked about gram staining and acid-fast staining. So capsule staining is our third type of differential stain. What are capsules? Capsules are protective outer structures that are made of either carbohydrates or proteins or a combination of carbohydrates and proteins that are linked to microbial virulence. Virulence is the ability of a microbe to cause disease, and it turns out that bacteria with capsules are usually more virulent than bacteria without capsules. So if you have something with a capsule, it might be more likely to make you sick than a bacterium that does not have a capsule. The thing about capsules is they also are not great at absorbing dyes, and they require negative staining methods. So they don't actually stain well at all, these capsules. So usually what you do first is you use some kind of negative acidic stain to color the background, and this is usually the slide. And then the capsules will appear as a clear halo. You don't use heat fixing in this case, usually because capsules are very heat sensitive. So if you use heat, it'll kind of destroy or change the shape of the capsule. So you don't use heat in order to preserve the capsule integrity, that structure. The combined staining method usually involves first a negative stain to stain the background, then some kind of positive basic stain to stain the cells. And what you see in the result are stained cells, a stained background, and an unstained capsule in between. This picture in our book shows us a capsule stain where you're staining the background, a negative stain, and you're supposed to be able to see capsules around the cells, which are really hard to see. So I pulled another example online where you can see the internal cells. They're outlined with a little circle, and then the halo outside of that. So I can see like a cell, and then the halo around it. That halo around it is the capsule. This one I like a little bit better, and we're going to use something more like this in our class, where you stain the background. Here it's kind of like a purplish. Then you stain the cells with a basic stain. Remember that background was the acidic stain. The cells are stained with a basic stain, and capsules do not stain. So you see that white part, that halo, is the capsule. So negative staining helps us visualize capsules by staining that background, but not the capsule itself, because remember, capsules are not good at absorbing most dyes. So the end result of most capsule stains is the background is stained with that negative stain. The capsule is unstained, so it's like a clear halo, and the cells we usually stain to improve contrast. And this is great to help us visualize a capsule clearly, because we're not going to heat fix. Heat fixing will distort the capsule or sometimes destroy it. And it's great for identifying encapsulated bacteria, especially to see if we have some virulent strains present. That takes us to the end of the first half of 2.4, and the next half will finish those differential stains.